Hello, my dear students, and welcome back. I know, exams are just around the corner, and this chapter, molecular inheritance, it can feel a bit heavy, right? But I promise you, trust me on this, it's one of the most important chapters. And believe it or not, it's actually one of the easiest if you just follow along with me. We're going to break it down so simply, you'll feel like a pro in no time. Ready to start? Let's do this. All right, to get our brains warmed up, I have a question for you. I want you to imagine something that's probably taller than you are, something 2.2 meters long. But here's the crazy part. It's hidden inside a space so tiny you can't even see it with your naked eye. And this secret, well, it's the key to everything about you. It's literally the blueprint of life. So, any guesses what I'm talking about? Yes. Can you believe it? 2.2 meters. That is just mind-blowing, isn't it? That's the actual length of the DNA that's coiled up and packed so perfectly inside the microscopic nucleus of every single cell in your body. So here's our game plan for today. It's super simple. First, we'll look at life's master plan, the DNA itself. Then we'll figure out how it makes perfect copies of itself. After that, we'll see how the messages get sent out from the DNA. And finally, how those messages are used to build everything. And we'll wrap it all up with a super quick revision for your exam. Simple, clear, and no confusion. Okay, first up, let's talk about DNA. The best way to think about it is like a master recipe book, or, the main blueprint for a huge building. It contains every single instruction for making you, well, you. Everything from the color of your hair to how your body knows how to fight off a cold. So, what is its official full name? Deoxyribonucleic acid. Now, don't let those big words scare you. All it really means is that it's a very long molecule, kind of like a thread, made up of smaller building blocks. And this thread is actually two strands twisted all around each other, forming that famous, really beautiful spiral staircase shape that we all know as the double helix. So the big question is, how on earth does that 2.2 meter long DNA fit inside that tiny little nucleus? Well, the cell has a really simple but brilliant trick. It uses these special proteins called histones. You can think of them like tiny little reels for thread. The DNA, which is negatively charged, wraps itself super tightly around these histone reels, which are positively charged. And this neat little package of DNA wrapped around a histone is called a nucleosome. How clever is that? Absolutely genius. Now, DNA isn't alone. It has a very important cousin named RNA. Think of them like two siblings, with totally different jobs. DNA is double-stranded and incredibly stable, which makes it perfect for its job, to safely store all the genetic information for a very long time. It's the master plan. RNA, on the other hand, is single-stranded and less stable, and that makes it perfect for its job, being a temporary, quick, working copy of just a small part of that plan. Okay, moving on. Let's see how this master plan gets copied. See, before a cell can divide into two new cells, it has to make a perfect photocopy of its entire DNA blueprint. This whole process is called replication, and let me tell you, the cell's copying machine is unbelievably accurate. Now, before we get into the nitty-gritty, you need to know about what I think is the single most important flowchart in all of biology. It's called the central dogma. It's basically the fundamental rule for how life works. Information flows from the master plan, the DNA, to a working copy, the RNA, which is then used to build the final product, a protein. Just remember this, DNA to RNA to protein. This flow is the key to understanding everything else. So what exactly is replication? In simple terms, it's just the process of making a perfect, identical copy of a DNA molecule. The cell has to do this so that when it splits into two, both of the new cells get a complete and correct set of instructions. And here's the really cool part about how it's done. The process is called semiconservative replication. Semi just means half, so as you can see in the picture, the original DNA helix unwinds. Then, each of those two old strands becomes a template to build a brand new one. And what's the result? Two new DNA molecules, and each one has one old or parental strand and one brand new strand. It's a super clever way to make sure the copy is always accurate. Let's break it down into 3 easy steps for your exam. Step 1, the helix unzips, just like the zipper on your jacket. Step 2, a super important enzyme, its name is DNA polymerase, comes along and builds a new matching strand for each of the old ones. And step 3, Poof! You have 2 perfect copies ready to go. So just remember, unzip, build, and copy. That's it. All right, on to our next section. You see, the cell doesn't need to read that entire 2.2-meter long blueprint all at once. That would be crazy. Most of the time, it just needs one specific instruction. Just one recipe to make one particular protein. And this is where our friend RNA comes into the picture. This whole process is called transcription. I want you to think of the DNA as this huge, precious master recipe book that's locked away safely in a library. And the library is the nucleus. Now, you can't take the book out of the library, right? So what do? You take a small piece of paper and you just copy down the one recipe you need. That little note you just made, that's the messenger RNA, or mRNA. So a really important question is, how does the cell know where a recipe begins and where it ends? Well, it looks for something called a transcription unit on the DNA. And this unit has 3 parts. There's a promoter, which is basically the start signal. Then there's the structural gene, which is the actual recipe that needs to be copied. And finally, there's a terminator, which is the stop signal. Start, recipe, stop. So simple. Okay, we've got our message, the mRNA. Now it's time for the final step. This is called translation. The mRNA note travels out of the nucleus and goes to the cell's kitchen, which is called the ribosome. And it's here that the recipe is finally read and the final product, the protein, is built. Now, the language that the mRNA message is written in is called the genetic code, and it has some really important features you need to know. First, the words are always 3 letters long. These are called triplet codons. Second, the code is a bit repetitive or degenerate, which just means that different 3-letter words can sometimes mean the exact same thing. It's also universal, so a word means the same thing in you, in a banana, and in a tiny bacteria. And just like any good set of instructions, it has a clear start signal and stop signals. And this table, this is the official dictionary. This is the tool the cell uses to translate the language of mRNA into the language of proteins. On one side, you have all the 3-letter codons, and on the other, you have the amino acid that each one stands for. And as you can see, there are 64 possible combinations in total. Okay, it's exam practice time. Let's put that dictionary we just saw to use. Here's a very common mRNA sequence. What I want you to do is pause the video right now, look back at the table, and try to decode the sequence of amino acids yourself. Come on, give it a shot. I'll wait right here for you. So, did you get it? Let's check our work together. The first codon, AUG, is always the start signal, and it codes for the amino acid methionine. Then we have UUU, which, if you look at the chart, codes for phenylalanine. Next up is UUC. Wait a minute, that also codes for phenylalanine. And this is a perfect example of what we meant when we said the code is degenerate. See? 2 different codons are coding for the same amino acid. Well done if you got that right. All right, we're in the final stretch now. Let's do a super fast, rapid-fire revision of 2 key terms that you absolutely, positively must know for your exam. I can almost guarantee these definitions will show up. First up, what is a gene? It's really simple. It's the functional unit of inheritance. It's just a specific segment of your DNA that has the instructions to make one final product, which is usually a protein. It's that one single recipe in our master recipe book. And second, what is a codon? It's that 3-letter word we talked about in the genetic language. It's a sequence of 3 bases on the mRNA that tells the ribosome which amino acids should be added to the protein chain next. And before we finish, I just want to leave you with this one thought. You are made of about 30 lakh crore cells. And every single one of those tiny, tiny cells performs this whole amazing process. Replication, transcription, translation, perfectly, all day, every single day, without you even thinking about it. You just have to learn it and explain it once for your exam. If your cells can do it, you can definitely do it. You've got this. All the very best.